Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Navaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Habib, and I'm here with JP. How are you, JP?
Doing okay. Glad to be back.
Yeah, it's been a while since we recorded for various reasons. Lots of travel, some challenges from a respiratory standpoint on your side. So if there is some coughing or some clearing of the throat, please do excuse that for both of us. I'm not totally 100% myself.
I've got that sexy gravelly voice going on. So.
It's all good. We'll do some Luther Vandross type stuff today. I love it. But we're excited about a new topic that we're going to be embarking on, which is with regards to aging, longevity, and the process by which our bodies over time can break down in terms of their optimal function and some of the associated conditions that are linked to this. And today we're going to get into the topic of cancer a little bit. So why don't we start off just basically talking about this concept of aging and these theories of aging, something that you and I have had many discussions about.
Yeah. So as we've talked about, there's many ways in which we can explain the phenomenon of aging. We can explain it through telomeres, shrinking of the ability of cells to reproduce with high fidelity their DNA as they divide and reproduce. Another is through the management of energy in the cell. And of course, the most important way we generate energy as eukaryotic cells is through our mitochondria. And if the mitochondria get damaged or degrade or aren't functioning properly through oxidative stress and other things, toxins, we don't function optimally at a cellular level, and that can lead to lots of problems. And we'll talk about that too. There are other theories, but really, I think those are two of the biggies. We'll get into other ones along the way through the season as we talk. But the topic that I've been really focused on recently is cancer as sort of a microcosm of aging itself. You know, we know that you're more likely to get cancer the older you get in life. Why is that? And there's multiple theories on that. And I think it's important for us to tease out how those theories might overlap, and maybe the truth lies in sort of a balance between the two. You know, one of the things I've always been sort of amazed by is when you've got two camps that sort of are fighting one another with respect to some idea or explanation of a phenomenon. A lot of the times you find that the truth is actually where those two groups meet or in the overlap. So I think maybe the truth behind cancer and maybe some tricks to how to treat it or overcome it will come from seeing where there's commonality between the two camps.
Yeah, I think where it comes down to specifically with cancer topic is there's a very strong contingent that believes that it's entirely a genetic condition and that there's a genetic variance or condition that drives the cancer process from a genetic standpoint. And we've got some other research that I think is pretty tangible and pretty strong, talking about the metabolic basis of cancer. And then somewhere in the middle, maybe like an epigenetic standpoint, is really where the truth lies. And there is probably both the genetic and metabolic container that drives this factor. And you hit the nail on the head. Why is it that as we age, as we become older, we have this increased risk of cancer? And this is across the board, right? Almost, unless there's childhood cancers, obviously those are not associated with aging, but they are associated with age being in a childhood type setting. But with the more common cancers that we're talking about with regards to aging and longevity, there is an increased risk simply just by the progression of time in your body. So what are the factors that might be contributing to this? So why don't we break down a couple of these concepts a little bit? Why don't we get into the genetic basis initially, and then we can talk about the metabolic side and combine it all after that.
Sure. And if you go to the National Cancer Institute or to the National Institute of Health and you ask people, okay, what's cancer? they'll recite the party line, which is that cancer is a disease of somatic mutation. What does that mean? That means that the genes within a given cell, within a somatic cell, not in a germ line, although that can happen too. That's when you're predisposed to it. But within a given cell, that there'll be a mutation of a gene. And that gene mutation leads to some dysfunction in the cell that dysregulates its growth. And maybe it's worthwhile explaining, what's a really good definition of cancer. And I personally like the definition of it's unregulated growth. It's a cell or a group of cells that have lost the ability to regulate their growth properly. And so to understand cancer, you really have to understand what does it mean to regulate growth properly? And that's a really important topic that we'll get to. But with respect to somatic mutation being the driver of cancer, the idea is that there's, again, a mutation that takes place, could be because of a toxin, could be because of radiation, could be because just bad luck. Although there's some reasons to believe that bad luck isn't such a great way to explain it. And I'll explain that through a paradox in 1 second. But that as a result of that mutation, the cell gains an ability to grow with some less regulation. And as a result, it grows faster or proliferates into a tumor or spreads throughout the body through metastasis. And you get unregulated growth that then leads to dysregulation of your whole system. Now, I said that it isn't such a great argument to say that it's just random. And there's something called Peto's paradox, which is really kind of an interesting thought experiment, if you will. It says, if it's just a function of random mutations taking place, then you would think that bodies or species that have more cells would have higher levels of cancer. And it turns out that that's not the case. That's the paradox. Do humans have more cancer or less cancer than animals that are smaller or much larger? So, for example, do elephants or whales have more cancer than human beings because they have, an order of magnitude or more cells than we do? And the answer is that they don't. They don't have more cancer. In fact, and they live just as long. I mean, whales can live for 60, 70 years or more. There's some whales that actually, barrel whales don't appear to have a defined life expectancy. They can live for hundreds of years, not more cancer than humans. So there's some evidence that part of the reason why that's the case is because they have multiple copies of some tumor suppressor genes that help cancerous cells, when they start to turn that way, to commit suicide and die, so they never actually end up proliferating.
A greater regulatory capacity, basically, with the genetic kind of built-in system or a kill switch.
Right, but it's interesting to note, and we're going to get into this also, it's interesting to note what that gene is and what that gene does. Because what I said was that it helped the cell commit suicide if it starts to get dysregulated. But it turns out that where that cell or where that gene functions, what that gene codes for, has a lot to do with mitochondria, has a lot to do with your metabolism. So maybe there's, that's the first hint that I'll drop that says it's not just your genes, it's your mitochondria and how they're functioning with one another.
Yeah, I think there's definitely an overlap that needs to be looked at here. And the fact that there's a genetic basis, but also we're going to get into the metabolic side of this, I think, with some depth, but understanding that it's not simply a genetic factor that's driving these conditions. And if we look at this across, as you mentioned, all these different species, it's not a genetic basis only. There's very clear links to other factors and other internal processes that are regulatory in their function, and that there is a regulatory component. And it feels like that regulation is really the key factor here. Like, growth is good generally, but uncontrolled growth or unregulated growth is bad. If we're not growing, the cells essentially would be dying. So they're not replicating, they would be dying off. So there needs to be some level of growth, but it needs to be regulated within a good bell curve amount of that growth. And if it goes too high, that's where we get into cancer, because the regulation is lacking. The regulation is where we need to really focus our attention from a therapeutic or preventative standpoint. So
Yeah.
I think we can get into that a little bit as well, but it sounds very much like that regulatory piece is often what's missing.
Yeah, and I think it's important to say that there's two phases of sort of healthy activity within a cell. You can be growing, that's sort of the anabolic state. And then there's what's referred to as the catabolic state, which is the opposite. Instead of growing, it's sort of a homeostatic state where it's healthy, but it's not growing. It's a much reduced activity level or a different level of activity. And we'll talk about the pathways within the cell, and these are ubiquitous. I mean, it's across every type of eukaryotic cell, from the simplest yeast and fungus all the way through, humans, elephants, and whales. So what I'd like to get across to people is that that regulatory control, when it's healthy, is what breaks down. And to understand how it breaks down and the consequences of how it breaks down, you really have to understand how it works when it's healthy. Because a lot of the cancer medications, especially the next generation of cancer therapeutics that are coming down the pike, the PI3 kinase inhibitors, things that are like rapamycin and the other rapalogs that are going after the what's called mTOR. We'll talk about that. We have talked about that. Those things are really important. Things that activate autophagy are really important too. But there's also a lot of really smart people. And I'll give a shout out to Thomas Seyfried up at Boston College, who's really been the pioneer in the field of metabolic explanation of cancer, looking at ways to treat cancer that go to that argument or that explanation for how cancer thrives and survives. And there's a lot of truth in what he has to say.
What are some of the specific topics and the specific findings that he's had there? And let's talk a little bit about this metabolic basis
Sure.
of cancer.
So what Thomas Seyfried, and I don't want to put words in his mouth, I'd love to get a chance to talk with him. I think he would be a wonderful guest for us if we can get him. What his argument is that if you, and this is experimentally verified, he said if you take a cancer cell, if the argument is that there's a mutation in that cancer cell's genes that's driving the cancer, then if you were to transplant that nucleus with the DNA from that cancer cell out of that cell and put it into a healthy cell, then that healthy cell should turn into a cancer cell. That doesn't happen. It's not what happens. However, if you take out the mitochondria from the cancer cell and you put it into the healthy cell, that cell very likely will turn cancerous. And by the alternative, if you take the healthy mitochondria out of the healthy cell and transplant it into the cancer cell, a lot of times that cancer cell will commit suicide and it will just leave. So there's a lot of reasons that's sort of the original thought line or reasoning behind, or one of the supports for his argument, that it really has much more to do with mitochondria. But just simply seeing that doesn't give an explanation of how it's working. So here's what I've gleaned is what his argument is. And I'll probably, in the process of saying this, start to hint at how those two things, the two different explanations sort of merge, which is that over time, if you're not healthy and there's oxidative stress and you don't sort of do what you can to support mitochondrial health, there will be oxidative damage and an increase in the production of reactive oxygen species. Now, reactive oxygen species are sort of what the name says. It's forms of oxygen and it can be in the form of a singlet oxygen, which is at a highly active state, or other molecules that have that reactive oxygen on them. So those are what we call free radicals, that they have the ability to damage structures within the cell. In fact, that's how our immune system, some of our innate immune cells, how they actually kill bacteria and other things. They utilize these free radicals and reactive oxygen species as sort of weapons to damage these invaders. But it happens.
The concept of phagocytosis is literally like we eat it, but we utilize these reactive oxygen species and free radicals to literally punch holes in it.
Exactly. So what's happening in the mitochondria that are damaged or under stress is that they start to produce more reactive oxygen species. They produce an excess of this toxin. Now that damages the mitochondria itself. So it can do it only for a short period of time without needing sort of an overhaul. But the other thing that does is it causes damage, not necessarily directly to the genetic sequence of genes in the nucleus, but what it does is it degrades the epigenetic marks or the DNA methylations, the histone modifications that we've talked about on other things. Those are the ways that cells control what proteins are expressed and what aren't. And when they get degraded as a result of that, then the cell starts to produce proteins that it's not supposed to. And that looks a lot like unregulated activity. And if the wrong set of proteins start to get activated, well, that leads to unregulated growth.
I think it comes down, interestingly, to what is happening in the mitochondria there and what we are providing to the mitochondria to do its job. And so let's talk about what the general knowledge kind of the mitochondria is, but what is potentially this underlying effect of the transplantation of mitochondria, of healthy mitochondria to a cancer cell versus the unhealthy mitochondria to a healthy cell. What's going on in that, and why maybe there's more to the mitochondria than simply being the battery or the energy powerhouse of the cell. Maybe there's something more to it.
Oh, for sure. So maybe even take a step back in time and talk about what mitochondria are. So a few billion years ago, when life on Earth was very simple, all that really existed were very primitive bacteria. And bacteria differ from the cells that you and I have are made up of, and whales and elephants and mice and the rest of them, and even yeast and fungus, differ because they're so simple. All they have is a simple loop of DNA, maybe 10,000 base pairs long, has a few dozen to a few hundred genes. And those genes are expressed and utilize, the proteins utilize energy only from what's called fermentation, or basically the chemical modification of sugars or other saccharides, polysaccharides, et cetera, simply by breaking them in half. It's called glycolysis, literally glyco meaning sugar and lysis meaning break. So you break it apart. And in the process of breaking those bonds, there's energy released that gets stored typically in the molecule of ATP, which is adenosine triphosphate. Basically, you got a base, you got a sugar, and next to that base, nucleotide base in the sugar, you've attached a bunch of phosphates. And the phosphates are energetic, so when they jump off and they bind to something else, they basically deliver an electric charge. Literally, it's like a battery, literally battery pack. Now, glycolysis is a very primitive way of generating energy and electricity, literally, for the cell. What happened about 1.5 billion, 2 billion years ago is that a new form of bacteria came along, and really two forms. One was like blue-green algae, and it had the ability to take that carbon dioxide and water that was present, and with sunlight, combine them, release a little oxygen along the way, and form sugar. Now that we know of, those little blue-green algae came to be called cyanobacteria, came to be chloroplasts. So that was the birth of plant life. Now, those chloroplasts were generating lots and lots of sugar. So that was really important. So it was those bacteria that could grab one of those and keep it nearby or keep it inside it became plants, and they had lots of potential energy that could be supportive, or energy molecules that could be supportive of glycolysis. But then there was another really complicated little bug that evolved that had the ability, through capturing a much more complicated chemical process, to take that sugar or other molecules that had carbons and oxygens and hydrogens on them and break them down to carbon dioxide and water, and all the way, not just split them in half, but that through a complicated process, take it all the way down to carbon dioxide and water. And those were subsumed into some bacteria, because what they could do was generate 10 times, 15 times as much energy for every single sugar molecule. And that gave so much energy to the cell, these bacteria, which were sort of lumbering creatures, just sort of floating there, not doing very much. All of a sudden, they were supercharged. And with all of that excess energy, they were able to become more complicated. They were able to build structures inside them. Those are the organelles. They were able to support a much more complicated DNA. Now, not just tens of thousands of base pairs, but hundreds of millions and billions of base pairs. I mean, our human genome, every cell in our body has 3 billion base pairs. And that's actually relatively small. There are lungfish that have hundreds of billions of base pairs. And sequoia and certain trees have hundreds of billions of base pairs in their DNA. The energy necessary just to replicate that is so vast that the only way that could be done is if you have robust, really robust mitochondria supporting that energy generation. Now, along the way, those little bugs that had the ability to do that became the organelles inside our body. So we're really symbiotes. I mean, every cell in our body, every eukaryotic cell that exists is really a symbiote. There's that original bacteria construct, but it has the mitochondria inside it doing all sorts of things, generating that energy. But along the way, the mitochondria became really important for other things. It's not just generating energy. They're not just the power plant. They're like the seat of government. I mean, we think of the nucleus as running everything, but the nucleus is dependent on the mitochondria for many things. Now, there's mutual dependence, because a lot of the original genes for mitochondrial health migrated into and are now part of our nuclear genes. So let me just explain that a little bit. Every mitochondria has its own genes. So every cell in your body has most of its genes in the nucleus. Most of its DNA is there. But it has each mitochondria in that cell, and sometimes there are dozens or even hundreds of them. Those mitochondria have their own DNA. It looks a lot like bacterial DNA, actually, but they have their own DNA. But a lot of the genes necessary for the mitochondria to do their thing are now have migrated into the nucleus, which is why I said before, when you think about cancer, genes that are cancer genes, and when they're mutated, you end up with a higher risk of getting cancer. Well, it turns out that those genes that do that are actually genes that are part of that mitochondrial function. So saying that it's all a somatic, mutation problem, yes, but those mutations are driving mitochondrial dysfunction, because if they're not being produced properly, if you don't get the right proteins, then the mitochondria are sort of like not able to function properly. Now, they're very robust, and it turns out that no one gene, even BRCA1 and BRCA2, which are very famous because of their relationship with breast cancer and other cancers, the number of people that actually get cancer if they have that gene is still only about 50-50. That means half the people who have that mutation don't end up getting cancer. Now, why is that? Well, it's because only one gene isn't enough to completely disable the mitochondria. If you can keep your mitochondria healthy, well, then that gene, yeah, it's causing problems, but it's not going to necessarily lead to cancer.
I think it's important to kind of restate just how important that micro symbiotic relationship is between how the mitochondria and the nucleus work one-on-one together. And the fact that they are on a micro level symbiotic, right? That the mitochondria produces energy so that the nucleus can have its genes then replicated and pulled out into proteins that allow for the cellular function to be optimal. And at the same time, the nucleus is housing certain genes that allow for the mitochondria to do its work really effectively. And this symbiotic micro relationship internally within the cellular level is huge. And I just want to even dig a little deeper. There's a philosophical thing that you've got here talking about the more energy that one can produce to utilize at a cellular level, the greater the complexity of the cell, right?
Sure.
And that you could almost go to like a philosophical conversation, which we're not going to go down that path today, but you could go into a full philosophical conversation about the importance of the transference and the conversion of energy from potential to usable forms. And if you have a massive usable form, then you create complexity because you're able to then specialize function so intensely. And that's essentially what's happened with the evolutionary process, the process of evolution, and how we incorporated this bacterial process into our cells, and all life that we know of outside of the amoebic, basic, even bacterial life that we know of has gone to this level of complexity. And with complexity comes confusion, comes some challenge, and comes some very unique little things that need to be optimized. And so this is really important to understand, I think, from not just a philosophical basis, but understanding that it's that energy conversion that allows for processes to occur effectively. And how efficient, or the machinery that's being used in order to allow for that to work. This is important because this is a difference between, you mentioned glycolysis and oxidative phosphorylation, two very, very different ways of producing energy with tons of different outcomes and potential negative or positive effects on one side versus the other. So let's talk a little bit about that energy piece and the efficiencies that we have with glycolysis versus oxidative phosphorylation.
Sure. The way you described it as sort of almost a philosophical thing, I think that's a really important perspective that a lot of researchers in the field maintain almost as a subconscious bias, that they recognize that in order to really understand any process within the cell, you have to think about it as a movement of particles of energy, if you will, or units of energy. And if you don't think about it from a metabolic standpoint, you haven't really considered how it all works. Because every process, every biochemical process that goes on inside a cell is technically an uphill struggle. Those biochemical pathways typically do not happen easily. They require the input of energy. They're not energy favorable. So life itself is a struggle. And so you need to maintain the production of energy in order to be successful. Thomas Seyfried talks about it, but it's just a truth that cyanide is a very, very quick way to kill something that's living because it disrupts oxidative phosphorylation. It disrupts the ability to create energy. Now, and I want to just spend one moment talking about this because it does relate to when energy imbalances become extreme in the cell, it leads to the cell committing suicide, which is actually a process that is most often triggered by the mitochondria themselves. The cell nucleus doesn't decide that it's time for the cell to die. It's the mitochondria that basically go through the process of releasing some of the molecules that they use to generate that energy. And when they do release those, that leads to a whole cascade of proteins working together to cause the cell to, in a very organized fashion, to expire and then presumably be replaced. That process takes place inside the mitochondria. We talked about the fact that there's this process, this complex chemical process called the citric acid cycle, or the Krebs cycle, that breaks down things like sugars all the way down to carbon dioxide and water. Along the way, that process is creating energetic molecules, usually typically NAD and FAD molecules that are basically holding on to a proton. And those activated molecules of NAD and FAD end up being used by a separate process, complex 1 through complex 4, that's sitting inside that mitochondrial membrane, inside the mitochondria, and basically making a battery. I mean, that's what the mitochondria really is doing. It's creating a battery. It's pushing the positively charged protons away from where the negatively charged electrons are, across a membrane, just the same way a battery has a positive terminal and a negative terminal. And then there's one way to connect them so that the current can flow. And literally, it's a flow of current through a little protein that sits inside that membrane, across that membrane, called ATP synthase. And so what happens, these protons come along and they go through that ATP synthase channel, the circuit's connected. And as a result, the ATP synthase just generates ATP. It just sits there generating ATP as this circuit is connected. Now, when that breaks down, or when there's not enough of the ATP to be generated, so you don't have the production lines are halted, then you get a buildup of charge, and that charge then leaks. Just the same way a battery has a leakage current, there's a leakage of charge. And that leakage leads to the creation of reactive oxygen species, and not in a controlled way. And that creation of those reactive oxygen species leads to all the damage within the mitochondria to cause dysfunction, but it also leaks out and causes problems for the cell. And I want to get into what that problem is, because what it does is it takes the cell that's a very, very fine-tuned, like almost like a Swiss watch, how it functions, and it starts literally putting new parts in and taking parts out, and all of a sudden the watch isn't functioning properly. It's not telling time well, and it might still look okay on the outside, but inside it's very dysfunctional. And getting into how that works ties us back to, okay, you've got the genes that are mutated that are making you predisposed to cancer, but what are those genes supposed to be doing? They're supposed to be keeping the mitochondria healthy. They're supposed to be keeping the mitochondria functioning properly. When they don't have the right genes, they don't have the right proteins, the mitochondria start to not function properly. The battery starts to leak. It starts to create reactive oxygen species and free radicals. Those damage the mitochondria further. And there's this other thing that's happening inside the nucleus that causes the cell to no longer function like it was designed to function.
I love that. I think it's important to understand that not only is the mitochondria literally functioning as a battery in creating charge and creating this, or shifting the potential energy of the macronutrients that you're providing to it and breaking it down and producing ATP and having this tool of transference of energy from one area to another, but it is a self-regulating battery. That if there is any mechanism or any driving factor that's causing the battery to not be able to function at a high efficiency, it has this self-regulating kill switch basically that says, send out the DNA, send a signal to the immune cells nearby, and let's get rid of this particular cell. That is so important to understand, because that self-regulatory capacity is a major missing piece when it comes to then dysfunction. It is, in fact, the missing piece when it comes to dysfunction.
For sure. It is.
right? Because that's where the dysregulation occurs. Because if you don't have that regulatory capacity from the mitochondria, then you don't get the signals to the immune cells. The immune cells can't come and phagocytize and apoptose the cells that are asking to be knocked out. And so there's a heavy link to the immune system here as well, but there is this self-regulatory capacity of mitochondria of every somatic cell that has a mitochondria as a part of it.
Yep.
So important to fully understand.
Nope, you're absolutely right. And I think the final piece, or not the final piece, but a really important next piece in the puzzle is something we spent a little bit of time talking about on a previous podcast, which is epigenetics. And it's worth now talking about how does a cell or how does a body like yours or mine come to be? We've talked about that extensively and how the immune system plays a role in development. But within the cell, you have initially what's called a pluripotent stem cell. That's literally when you're conceived. You've got the omnipotent one, pluripotent stem cell, which continues to divide and divide. And as it goes through that division process along all those different branches to ultimately create the trillions of cells that make you or me up, what happens is there's a differentiation process. Initially, all of the DNA that you've got is needed. But as a liver cell, as you're along that branch that's going to go down to the leaf that's a liver cell, you don't need the acorn cell. You don't need the instruction manual for making bark. You don't need the instruction manual for roots because you're going to be a leaf. So what happens is, in order to reduce the amount of complexity that cell has to deal with, it starts to lock away all of the genes that are no longer needed. And it does that through epigenetics. We've talked about that before. There's multiple ways that it does it, but two of the really big ones, and for our purposes right now, really important ones, are DNA methylation, which basically means sticking a methyl group onto one of the bases, and it's actually a two-base sequence of C's and G's, putting a methyl group onto that C of the CG sequence, and as a result, that sort of locks the DNA away, so it doesn't open up and can't be copied. I think about it like if you're building an airplane. Okay, if you're going to build a 747 with 1 million parts, you got a team of people, scouring over the structure, building it. Those, we can think of those as sort of like your tissue resident macrophages. They're doing all the work. Okay, and then you've got the pieces, and the team that's going to build the landing gear doesn't need the instruction manual, the portions of the instruction manual, which might be 1 million pages thick. It doesn't need the section talking about the cockpit. It doesn't need the section talking about the engine. It needs the parts talking about the landing gear. So do they want to carry around loose pages that have cockpit stuff in it? And no, they don't need that. So they've got their own miniature form of that. Now, in the case of the cell, they still have to carry it around, but they carry it around locked down. They lock it down through these methylations, both of the DNA, which we just talked about, as well as the proteins called histones that the DNA is wrapped around. That's the histone modifications. And that can be methylations or acetylations or other chemical modifications. But basically the same thing is the case. It's locking it in so that the DNA either can't be opened up and reproduced, or in some cases, it's actually the opposite. It's actually methylated so that gene will stay open and always be available. Think of it like almost like putting a wedge on a door that's closing, keeps it open. And those chemical modifications of the DNA allow the cell that is going from that stem cell state, where it could be many different things, down to the very specific, hepatocyte or a myocyte or a neuron or any other type of cell. It's going through that differentiation process, and those epigenetic marks are in place to prevent it, most of the time, from that cell producing proteins and expressing genes that it doesn't need to. Now, we talked about unregulated growth. One of the hallmarks of cancer is all of a sudden that cell is producing proteins that it's not supposed to. It's ceasing to be the specific cell that it's supposed to be. It starts to become sort of something else. In fact, there's even tumors. There's a form of tumor that once it starts to grow, it starts to produce, it's kind of gruesome, but it can produce teeth. It can produce like whole parts of organs inside the tumor because there's a de-differentiation process that then starts to differentiate again into something that obviously shouldn't be there. I mean, you don't want to find inside a tumor, other body parts that are sort of starting to form. It's kind of gross. Think about it.
Yeah, you don't want to find a pancreas and a spleen, right? Like it's not what you want to see.
No.
Yeah.
But that's a function of the fact that as part of the cancer, all of a sudden those other genes are being unlocked. Now, how does that happen? How does that unlocking happen? Well, it's those reactive oxygen species that are being created in prodigious quantities by the mitochondria that are dysfunctional, that are under oxidative stress. So if you go back upstream, again, you got the genes, maybe you got some mutations. What are those mutations of? There's mutations of genes necessary for proper mitochondrial function. Now, the mitochondria aren't functioning properly. They're under stress. Now, that can happen because of those genes, but it can also happen because of toxins. It can happen because of obesity. It can happen because of a whole host of other things. Stress in your life, infections. There are viral infections, and there are infections that lead to cancer. And there's lots of cases of cancer that arise as a result of a trauma, okay, where there was a need for growth, there was stress. And what happened was the mitochondria started producing lots of reactive oxygen species as a result. And those reactive oxygen species unlock the epigenetic locks on the DNA, alter the epigenetic landscape. And all of a sudden, you've got different proteins being generated inside that cell. Now, most of the time when that happens, the cell is still functional enough that it says, okay, I'm dysfunctional. Mitochondria, release your cytochrome C and your DNA. We're gonna send the signal out to the immune system. We're gonna expire, and we're gonna go through apoptosis, and we'll get replaced by a healthier cell. Okay, all of that is normal. It happens probably hundreds of times a day in every person on the planet. So that's not a problem. It only becomes a problem when the wrong set of genes are unlocked. And it appears to be random. The wrong set of genes are unlocked. The apoptotic process is avoided, or the damage to the mitochondria is sufficient that they can't do their job of triggering apoptosis. And now you've got unregulated growth. Now, that is how you get to a cancer cell, or at least a very logical explanation for how you get to the cancer cell. Now, again, the two camps are one saying, oh, it's all about genetic mutations. Okay, maybe that's a part of it. We're going to keep that in the back of our heads. The other group is saying, no. It's caused by the mitochondria being dysfunctional. And the mitochondria dysfunction leads to reactive oxygen species that unlock the DNA and cause all of these proteins to be created that shouldn't be, and the cell stops being specific, and it gets unregulated growth. Yeah, that's a really good explanation, actually. I like that. Okay, and there's lots of evidence that mitochondrial dysfunction is going on inside these cells, and we should talk about the Warburg effect. But if you piece those two things together, if you knit them together into an explanation for how both sides are right, both sides are part of it, but the level of mitochondrial dysfunction doesn't have to come from a genetic mutation. You can have higher, and we see higher levels of cancer among people who have lots of oxidative stress, who've put their cells in a bad position, either voluntarily or involuntarily. Obviously, I don't mean to there's no moral judgment here, but when the mitochondria are in a distress state, that oxidative stress state, they're going to create more reactive oxygen species. Those reactive oxygen species have the ability to disrupt the epigenetic controls, and you're going to see degeneration. You're going to see a slipping away from being a young, healthy, very specific and active cell into something that's doing a lot of things it's not supposed to. It's distracted by a lot of other proteins that are it shouldn't be producing.
I'd love to just summarize kind of my thoughts here, because we're talking the whole process, which I love. So we have these pluripotential or omnipotential cells initially as stem cells when we're first in mother's womb. When there's conception, we have like the most potentiality, and it's just a massive amount of energy that comes together, and we have these cells that have the potential to become anything in reality. And what slowly happens is genetically we have these blueprints of what this person could potentially be. And in this blueprint are the manual as to how each of these different areas of this potential person could work. And as we lock down and we eliminate the potentiality, we become hyper specialized. And it's epigenetic factors that trigger this hyper specialization. So it's specific epigenetic and specific environmental factors that drive a hepatocyte to become a hepatocyte in the liver. And that hepatocyte, that liver cell, has a very particular set of functions that it needs to produce. And so in the manual that the hepatocyte has in its nucleus, there's a few pages in that owner's manual that are completely open with bookmarks, methylation, whatnot, and histone modification. And then we've got a ton of that rest of that manual locked away. And what happens occasionally is there's dysregulation, as in some of the locked pages become unlocked for whatever reason. And if they become unlocked, it's not a guarantee that those unlocked pages are going to become a regrowth and go backwards negatively towards potentiality. There needs to be a certain set of circumstances that allow for that cell to revert. And the major circumstances there are the presence of excessive levels of oxidative stress linked to excessive mitochondrial dysfunction. It's this combination of factors that as we grow optimally, we go into hyperspecialization and we remain in that hyper-specialized state with great regulation from that cell itself, from the mitochondria nucleus communication that's occurring effectively, and regulatory function from the outside, from the immune system, making sure that the cells that are specialized continue to do only that specialized work. And when they don't, that results in potential of getting rid of that cell, or the need to have that cell either sacrifice itself. And on a regular basis, that is occurring because these mistakes can be made every so often at the genetic level. When the mitochondria are dysfunctional and when the immune system is incapable of regulating that from an external standpoint, that's when the cancer increases, or the risk of that cancer increases.
And that's where your unregulated growth starts. And that's how the next phase of this conversation is about, well, what is growth? What is regulated growth look like? Because we do need to, cells need to reproduce, cells need to be replaced, cells need to grow during development. What's happening to drive that? And how do we control that? So to understand that, let's go back a couple of billion years, okay? We've got those, before the mitochondria, before the chloroplast, you had bacteria, and they were very, very simple, okay? And when they came across nutrients, they took those in and they grew, they reproduced in order to survive and, and to perpetuate their species. They needed to really utilize every opportunity to grow. Now that meant that they didn't regulate their growth. They just said, listen, we're on all the time. If there's nutrients, we're growing. Okay. And we'll use up all of the nutrients, independent if it means that we drive the nutrients to zero and then we all die. We're just going to grow. So with that unregulated growth trigger associated with fermentation, which is all they did at that time, was just glycolysis, you had this sort of, I'm going to call it sort of the ancient bacterial mode, which is when I'm in fermentation mode, I'm gobbling up every bit of nutrients I've got and I'm going to grow. Okay? I'm in growth mode. What happened when the mitochondria came on board and there was a lot more energy? They started to develop, one of the complexities of the system was they developed a more sophisticated control mechanism. And it turns out that control mechanism is sort of, it's got two sides, like the yin and the yang. You've got the side of growth, pro-growth, which is typically referred to as the PI3 kinase AKT mTOR axis or pathway. And then on the other side, you've got the AMP kinase and autophagy. And to describe those two things, because that's a word salad there, or a letter salad. What you've got on the growth side, the anabolic side, is a sensor or a collection of proteins that have sensors on them. They can sense temperature, oxygenation, hydration levels. They can measure amino acid levels. They can measure energy levels. And that complex is based around the molecule called mTOR, which is called the mechanistic target of rapamycin, which is interesting that they named the molecule after a drug called rapamycin. But what that molecule does in its complex is trigger growth through several different pathways. It promotes protein expression. So at the nuclear level, it's causing you to take all those genes that are supposed to be produced in that cell, and maybe some of the ones that aren't, and it causes them to be produced. It produces new structures inside the cell. It tries to reproduce. It creates the conditions for reproduction, going through the cell phases. On the flip side, you've got your AMP kinase. That's when mTOR is not sensing a sufficient amount of amino acids or other things. When you're in starvation mode, this is the advance, if you will, over that fermentation, ancient, grow all the time mode that the bacteria without these controls was doing. This is the ability to say, hey, when those nutrients are low, when we don't have enough, we're going to start to recycle things inside the cell. And so literally what AMP kinase is driving is this process of creating, it's like a garbage bag inside the cell. All the broken things, all the things that it doesn't need anymore, go into the bag. And then the bag merges with something called a lysosome, and all the enzymes inside that lysosome get released onto that bag of garbage, if you will, or recyclables. And literally, it breaks them down to the nutrient level, to the individual amino acid level or sugar level or whatever. And then that gets released back into the cell. So the cell is basically taking its garbage and turning it back into what it's going to use as its fuel and food. That process is critically important. And the reason why I say it's not just energy efficient or anything like that. It is the way the cells clean house, because it's not random what goes into that bag. What goes into that bag are mitochondria that are broken down. It's proteins that are misfolded or structures like other parts of organelles that aren't needed anymore. So all that superfluous stuff and damaged stuff gets put in the bag and then rebuilt properly. So if you've got a cell that's got a dozen mitochondria and 3 of them are damaged, and you never get yourself into that recycling mode, then you're in oxidative damage state a lot. And that's going to keep producing reactive oxygen species and damage the cell further. If, however, you shift out of growth mode into the autophagy state and you drive through AMP kinase, what happens is you get rid of those 3 mitochondria that are broken. It's actually called mitophagy. And you replace them. Over time, you'll replace them with 3 more mitochondria that are made from the ones that aren't broken down, that aren't damaged. So if you want to sort of perpetuate youth, you want to minimize the damage that's going on, you want to shift into that state regularly. You don't want to spend your life, always in growth mode. Now, how do you do that? Well, it's one of the things you and I have talked about a lot, which is get yourself into really good shape. Eat properly. Eat less, fewer things that are inflammatory, fewer things that are sugar-based, fewer things that are carbohydrate-based, and feed your cells what they need in order to shift into that state more regularly, and just generally how to use your mitochondria in the least destructive way. And it's a whole separate conversation about that, but it's important for let's dive into it just for a second, okay? One of the phenomenon that has been noted and has been known for over 100 years was found by Otto Warburg. One of the things that Thomas Seyfried talks about a lot is this Warburg effect. And cancer cells have mitochondrial dysfunction. So they don't create their energy through oxidative phosphorylation. They rely almost exclusively, if not totally exclusively, on glycolysis. Now, glycolysis is sugar. Okay, so you're breaking down sugars. Okay. Now, it turns out that you can also break down sugar-based amino acids like glutamine and glutamate. Okay, but mitochondria can utilize other things. They don't just have to use sugars and glutamine. So if you get rid of the glucose, minimize your glucose levels. If you feed yourself only the things that the mitochondria can use, but you can't use for glycolysis, then the cancer cells will literally starve. If you've got lots of sugar in your system, the cancer cells can live. If you have no sugar in your system and you minimize glutamine levels, you can actually drive the cancer cells to literally starve to death because your mitochondria have the ability to use fatty acids. So
this is literally the therapeutic basis for ketosis and ketogenic diets as a major driver of anti-cancer therapeutics. Another one that I want to kind of bring up here that we didn't mention much, but it makes a ton of sense, is fasting, which drives a very similar response, right? Fasting is one of those very efficient ways to turn off mTOR and get us into that situation where it's almost like a controlled burn, right? Yep. If you ever imagine, or if you ever see farmers doing a controlled burn on a portion of their land, it's simply because there's diseased or there's broken down function of the crop that they're producing. And so they burn a section of it to literally break those crops down into their components, get the nutrients, the broken down minerals or the broken down amino acids down into the soil again, regrow that using the minerals and whatnot you've broken down in the soil to then regrow and redo what you're doing. It's the reset button or the power off and power on your computer when things aren't working because there was a glitchy situation going on. It's the same exact idea, but in a biochemical function.
It's what Mother Nature does with forest fires. I mean,
exactly.
What's the healthiest area of the forest? It's the one that had the burn last year.
Yeah. The
That's
most recent burn,
that's the area
exactly.
that's growing the fastest. What you don't
Yeah.
want, I mean, yes, we love old growth forests and all that. That's really important because they've gotten into a cycle that's very healthy. But
Yes.
if you need to preserve those and prevent forest fires, scorching, hundreds of square miles, you need to do controlled burns, much the
Yeah.
same way the farmer does.
Yeah. That's where we can cycle into ketosis and out of ketosis and cycle with fasting on and off. And the therapeutic basis for some of these clinics out there that are doing like 21-day fasting, and you're like for cancer therapeutics, and they have phenomenal results with patients that are willing and capable of doing that work.
Yeah.
And they're able to actually have a significant positive effect on these people's lives once they realize that there is a strong metabolic basis to what we can do therapeutically, because we can't physically go in and change the gene or the gene mutation that's occurred. But what we can do is stimulate the regulatory capacity of the body. And the regulatory capacity is what's driven through that controlled burn function, that fasting or that ketogenic state for a short period of time. And that's what we need to really focus on, because what we have the capacity to do in a practical sense is simply to send out very particular regulatory messages. Regulation of growth is the key here. And so in a practical sense, that's what we're always talking about, because it comes down to the immune system being capable of regulating growth as effectively as possible. The presence of these tissue resident macrophages, if they are given the right tools to do their jobs and they get the right stimulation from the vagus nerve and through acetylcholine, through the chat cells that are coming through, those cells remain in a very strong regulatory function. Versus if they're in a heavily inflammatory state all the time, they're creating more reactive oxygen species. They're creating more of these oxidative stressors that will result in further degeneration and lack of regulation of the growth that's occurring locally in those tissues.
Yeah, and that's the extracellular piece, but there's the intracellular piece, which is why one of the most fabulous things I've learned over the last decade is the fact that it's actually the same message. It's that acetylcholine that you were talking about with respect to the macrophages, but that's on the mitochondria also.
Mitochondria as
The
well.
same message. I mean, if that's not a blinking neon sign with a big arrow saying, "Hey, this is important," I don't know what is. But what you were talking about extracellular, I'm also talking about intracellularly, about the mitochondria. That that therapeutic ketosis that you were mentioning, that's actually one of the really important components of this whole metabolic approach to treating cancer. Again, your glycolysis, the cancer cell has to rely on something other than the mitochondria for generating energy. And therefore, it can only rely on glycolysis or glutaminolysis, which is basically using glutamine.
Yes.
Now, the challenge is, because it'd be great, let's just starve every cell of glucose and starve it of glutamine. You can do the first, okay? We can literally just go into therapeutic ketosis, get our levels of glucose down to negligible levels, okay? But you can't get rid of glutamine, because glutamine is an amino acid that your body makes. There's no way to get around it. To a certain extent, you can say the same thing about glucose, because your liver can produce glucose, and it does. Okay, so you're not going to really get glucose down to zero, but you can get it down pretty low, because if you provide the cells with an alternative energy source, the healthy cells with an alternative energy source, which is fats, healthy fats, fatty ketone bodies. If you deliver to your cells through diet, if you deliver to your cells enough of the fats that it can use for energy in the mitochondria, then the cells won't be asking your liver to produce glucose, because that's how it happens. Your cells produce signals that tell your liver, hey, we need more energy. We need more energy. And the liver's it can do two things. It can make more fats. It can go through, gluconeogenesis or liponeogenesis. if your liver is being tasked with creating energy, it's going to produce glucose and it's going to produce fats. If you provide the fats, if you provide the energy to your body in the form of fats, then your cells won't starve. The healthy ones won't, but the cancer cells will. So again, the idea behind what Thomas Seyfried and other people who are promoting the metabolic approach to treating cancer is get into a state of therapeutic ketosis, provide the cells with fats that they need, the healthy cells, starve your cells of the cancer cells of glucose. And there's a few other things that play into it. Hyperoxygen levels are good too, hyperoxygenation. And there are ways to block glutamine, but you can only do that for a short period of time because it can actually damage your immune system. But like on the level of hours, there are some medications that will block the production of glutamine.
Seriously.
And so they say it's the press pulse method. You're pressing glucose all the time, and you're pulsing every couple of days, once a week or so. You pulse, blocking glutamine for a period of time, load up on your oxygen levels, and the cancer cells just die away.
Can't handle it. Yeah.
Can't function.
Absolutely. I want to be respectful of time, so
sure.
Let's take a moment to wrap up just everything we talked about. There was a lot in here, understanding kind of the development of the cell, the regulation and the regulatory capacity of internally within the cell, the mitochondria having that very important regulatory capacity, the symbiotic relationship that's occurring internally within the mitochondria and the nucleus working together synergistically to have a good level of growth, but also regulatory capacity for each to function at a good level. And what happens when the system breaks down, when the regulatory capacity breaks down, and understanding that there are moments where we need that control burn, where we need to shut things down and actually just reset or restart. And that's where these therapeutic diets or metabolic based therapeutic approaches to treatment of cancer come in, and how they are as successful, I would say, as they tend to be. So really important, really interesting discussion. Any last points you have that you want to bring up?
Yeah, I think we started out by talking about the fact that, when you hear two different people arguing that they have the explanation for why something is occurring, why some phenomenon is happening, and there's truth on both sides, then the chances are that they're just each giving just a piece of the story. And what we tried to knit together here was the somatic mutation theory of cancer isn't entirely wrong. It's got some really important things to say. One of the things that gets lost in the mix is that a lot of those cancer mutations that get talked about, what do they do? What are those proteins supposed to be doing? What happens when they're mutated and not doing what they're supposed to do? A lot of them, a tremendous number of them, have to do with metabolic function. So the metabolic explanation of cancer, which is all about oxidative stress on the mitochondria leading to elevated levels of reactive oxygen species for long periods of time, leading to not just inflammation, but that's really important, but leading to a loss of epigenetic control, that leads to the dysregulated growth, the expression of proteins that the cell shouldn't be producing and becoming cancerous. And that the two pieces go together, that there's lots of ways that metabolic dysfunction can occur, but one really important way is if the building blocks of those mitochondria and the proteins that they need to function properly are mutated. So is it 100% one or 100% the other? No, but if you put them together, you can see how this pathway is probably the dominant reason or mechanism by which cancer occurs. And by understanding that, then you can treat cancer with a metabolic approach.
I love that. Thank you so much for this awesome discussion, for breaking down this concept in such simple terms for everybody to understand. For everybody who made it to this point in the podcast, thank you so much for listening. And we wish you all wonderful health upgrades throughout your life. Enjoy.